you so much, Sayun, for inviting me to give this plenary. It's a real pleasure and a tremendous honor to be with you to spend the next 45 minutes or so to tell you about some of our work, some of the work going on in my laboratory. I always love coming to Korea. It's one of my favorite places to visit. So just to cover some of the background and the basics. This is the pathway that I have been preoccupied with for, over 30 years now, since I was a postdoc, which is the PI3 kinase AKT signaling pathway that operates in all cells and tissues in normal physiological signaling. As most of you, I suspect, know, this pathway responds, as all pathways do, to extracellular stimuli, whether they be growth factors or hormones or neurotransmitters, ligands that engage receptors at the surface, which through a complex set of mechanisms culminate in the activation of a family of enzymes collectively known as the PI3 kinases, so called because they interconvert a minor membrane phosphoinositide, a lipid, PI45P2, into a second messenger known as phosphatidylinositol 3,4,5 trisphosphate. This is the most negatively charged lipid in the cell. And that immediately tells you something about its function. This is generally not a structural lipid. It is not required for the integrity of membranes. It is a signaling molecule. There exist 3 AKT isoforms in mammals. They are distinct gene products that reside on different chromosomes, but with a very similar modular structure, as depicted here, and a very high degree of identity, which for the first 30 years or so of work led most people, including myself, to believe that these had largely redundant functions in both normal cellular physiology and in disease. But as was first recognized by Maury Birnbaum, who made the very first AKT1, AKT2, and AKT3 whole animal knockout mice that showed very distinct and non-overlapping phenotypes, we now know that AKT1, 2, and 3 have non-redundant, non-overlapping phenotypes in both normal physiology and in disease. For example, AKT2 knockout mice develop an insulin-resistant and diabetes-like phenotype, which you don't see with the AKT1 or AKT3 knockout mice. They have other distinct abnormalities, although all are viable, developmentally viable. And through the work of my lab and many other labs, we also know that AKT1, 2, and 3 have very distinct activities in pathophysiologies such as cancer, where AKT1 and 2, for example, have distinct roles in modulating cell migration, cell invasion, and metastatic dissemination. Now, going back to the wiring of the signaling pathway, as illustrated in this diagram that I've taken from a review that I wrote with my friend Brendan Manning about 3 or 4 years ago, covering 25 years of research in the field, once activated by this complex mechanism, AKT transduces a signal to a large number of substrates. But as we argued in this review, AKT transduces a signal to what we argue are 3 evolutionarily conserved multifunctional nodes comprising 3 very important substrates, 3 very important gene products: the forkhead BOXO or FOXO family of transcription factors, the glycogen synthase kinase 3 enzyme, and the TSC2 protein of the tuberosclerosis complex. All 3 are multi-site phosphorylated by AKT, and somewhat counterintuitively, at least at the time, phosphorylation by AKT serves to inactivate their function. Now, you would think for an oncogenic signaling pathway, phosphorylation would serve to activate, but in fact, for many AKT substrates, their phosphorylation serves to inactivate their function. For example, phosphorylation of FOXO by AKT serves to retain it in the cytoplasm, excluded from the nucleus, thereby terminating transcriptional activity. And the reason this pathway has gained so much attraction from the basic science community, from the drug development community, is because it is the most frequently deregulated pathway in all of cancer. In aggregate, if you query TCGA datasets for a number of the key rate-limiting gene products that participate in PI3 kinase AKT signal relay, including PI3 kinase itself, AKT genes, some of the substrates, the tumor suppressor PTEN, in aggregate, and you query for mutations, somatic mutations, gene fusions, gene amplification, copy number gains, or deep deletions in tumor suppressors, you find that in aggregate, this is the most frequently genetically deregulated pathway in all of cancer. I will turn my attention now to AKT inhibitors. Why? Because AKT inhibitors to date have not yet been approved. Yet. A whole number of drugs have been developed that are exquisitely potent and exquisitely specific. These are some of the drugs that are under development. So the two lead compounds in the clinic are ipatasertib, a Genentech compound, and capivasertib, an AstraZeneca compound. These are catalytic ATP competitive inhibitors with remarkable potency and selectivity, even for ATP competitive inhibitors. There's a whole bunch of other ones that are also in clinical development, but probably will not make it. So what are degraders? Most of you probably know, but it's a very cool pharmacology where you have two, these are strange drugs. But there's a very clever technology that was first devised, iterated by Craig Crews and his colleagues at Yale, where you have two warheads, they're called. One is a ligand that engages the E3 ubiquitination machinery. The other is a ligand that engages your protein of interest. Okay? And then they're hooked up with a linker. Okay? And the idea is you throw these onto cells, and the degrader will bring your protein of interest, in this case AKT, in proximity to the E3 that the ligand will engage. And of course, this will then target your protein of interest to the proteasome, because it's an E3 ligase. It will ubiquitinate your protein of interest. The proteasome will degrade it. And now, instead of having catalytic inhibition, what you have is wholesale protein degradation. That's why these are called degraders, or proteolysis targeting chimeras. And so through some very clever medicinal chemistry and chemical biology, we made the very first in-class AKT degrader, and we published this a few years ago. I'm not going to tell you about it because what I'm going to show you is our second in-class AKT degrader. We published this in Cell Chemical Biology. If you want to scan this, it'll take you to the PDF. It was very potent and selective. It degraded AKT. But we noticed both in cells and in vivo, it had relatively slow kinetics. It took quite a long time to get profound quantitative AKT degradation. So the chemical biologists went back to the hood and devised a second degrader, which we hoped would have better, improved kinetics. And I should say that for our first in-class degrader, we used ipatasertib, GDC0068, as the ligand to engage AKT, and we used cereblon as the E3 ligase. Actually, we used an IMID, lenalidomide, to engage the E3 ligase cereblon, which is a very common strategy. And so we made a second generation degrader, which also engages using the AKT inhibitor ipatasertib, but this time we used a ligand that engages VHL, the von Hippel-Lindau E3 ubiquitin ligase. And these are all unpublished, although this paper is technically in press and will be coming out hopefully soon in Science Signaling. And here is our second generation, and here's our first generation inhibitor. You can see that you put it onto cells in a dose-dependent manner. You get degradation of AKT at nanomolar concentrations. And you can see that it has improved degradation activity towards the first generation degrader, and it is much faster. Move on to the second part. This is all published, but Seyoun wanted me to present this, so I'm going to present it because it's one of the things that I'm perhaps most proud of in my career. And that is a paper that we published about a year ago now, which deals with the reprogramming of cancer metabolism by the PI3 kinase pathway. And the pathway that I'm going to focus on is coenzyme A. Okay? Coenzyme A is the major, I would argue that coenzyme A is probably the most important cofactor in all cells and tissues. There are others. And it's a universally conserved mechanism as a carbon shuttle. All right, it shuttles carbon. It is functional in obviously the mitochondria for the TCA cycle, at least when coupled to acetyl groups as acetyl-CoA. It is required for fatty acid biosynthesis, beta oxidation, and perhaps what you're most used to is the modification of proteins by acetyl groups. For example, histone acetylation. The entire chromatin code is driven by the post-translational modification of histones by acetyl groups. All of this derived from CoA and acetyl-CoA, which is fueled by glycolysis pyruvate to generate acetyl-CoA. And so this project began many years ago by a remarkably talented postdoc in my lab, Christian Dibble, when he did a very simple metabolomics experiment, steady-state metabolomics experiment, taking standard epithelial cells, treating them with insulin, which activates PI3 kinase, pre-treating them with a potent PI3 kinase inhibitor, and then doing steady-state metabolomics. And what you get are metabolites that change according to what has been published by many labs over the last 10, 15 years. That is to say, glycolytic intermediates in the pentose phosphate pathway, many intermediates in nucleotide metabolism. But there was one metabolite that we didn't even recognize by name, and that is pantothenate that accumulated in cells treated with PI3 kinase inhibitor. So what is pantothenate? It's also known as vitamin B5. And as Dr. Kim presented this morning, this is one of those curious and unusual pathways. It's very linear and simple, relatively simple as metabolic pathways go. Begins by cells taking up vitamin B5, pantothenate, and there is the input of cysteine, ATP, the AMP moiety of ATP, to generate what this is, which is coenzyme A, cysteine moiety, pantothenate. You take cells where we've engineered them with an oncogenic AKT mutation that is hyperactive. Okay? In the absence of insulin, these cells constitutively make CoA and acetyl-CoA. You treat, and this is in a doxycycline-inducible manner, you treat them with a catalytic AKT inhibitor, you completely block this constitutive production of CoA and acetyl-CoA, and rapamycin generally has no effect, arguing that this is downstream of AKT, but not as mTORC S6 kinase signaling. And the enzymes involved, there are 4 enzymes, the pantothenate kinases, and these 3 other enzymes. And if you scan the protein sequences of these enzymes, and if you scan phosphoproteomic analyses, you find that PANK1, PANK, and PANK4 in particular have high fidelity AKT consensus motifs that will be predicted to be phosphorylated by AKT. That are not found in any of the other enzymes that are required for coenzyme A biosynthesis. So the experiments then become very simple. You can show that PANK2 and PANK4, but not PANK1, are phosphorylated, as shown here, in response to insulin, and here, and this is blocked by a PI3 kinase inhibitor. We don't know if PANK1 is phosphorylated. It could simply be, even though it has a very good site, it could simply be that this antibody does not recognize this site. So we ultimately came up with a model in which cells stimulated with insulin activate PI3 kinase AKT. PANK4 is a suppressor of this pathway. Phosphorylation by AKT relieves this suppression, allowing flux from B5 to make CoA. We think PANK2, which we've studied a lot less, is a positive signal which initiates this mechanism. And now you make CoA. To get all the way to acetyl-CoA, you need an extra step, and you need ATP citrate lyase. Okay? ATP citrate lyase is also regulated by phosphorylation. What is that event? Guess what? It's also regulated by AKT. How do you get citrate for ATP citrate lyase to work? From glucose. How is glucose taken up from cells? Through glucose transporters that are regulated by AKT. That fuels glycolysis. How is glycolysis regulated? By AKT, both transcriptionally and post-translationally. Every step in this pathway has evolved over time to be sensitive to insulin and the regulation of PI3 kinase AKT to ultimately fuel acetyl-CoA, which is required for TCA cycle, histone acetylation, de novo lipid biosynthesis, and multiple other, and ultimately in aggregate, mechanisms that control cellular proliferation, which are altered in rapidly dividing tumor cells.